Hey, welcome to the Explainer. Today, we're going to do something really cool. We're going to decode the very language of life itself, the molecular code that literally connects every living thing on this planet.
I mean, have you ever really stopped to think about what you have in common with, say, a tree, or a mushroom, or even the tiniest little bacterium? It all boils down to one huge question. What are the fundamental building blocks of all life? Well, the answer is hiding in plain sight, inside these incredible compounds called biomolecules. And for this explainer, we're going to zero in on the big three, the ones that build our structures, provide our energy, and most importantly, write our genetic code. Basically, these are the molecules that build, power, and run the whole show. All right, first up, let's meet the real workhorses of your cells, proteins. Seriously, if your cell was a giant bustling city, proteins would be the workers doing pretty much every single job you can imagine, from construction to transport to communication. So what are these things actually made of? Well, proteins are what scientists call heteropolymers, but, a better way to think about it is like this. Imagine you have an alphabet with 20 different letters. You could create an almost infinite number of words, right? That's exactly what life does, using its 20 standard amino acids to build every single protein imaginable. But here's the secret. A protein's real power comes from its shape. It all starts out as just a simple, straight line of amino acids. That's its primary structure. Then, that chain starts to twist and fold into simple patterns, like coils and sheets. And finally, the whole thing scrunches up into a super complex 3D shape, kind of like a tangled ball of wool. This final, tertiary structure is absolutely critical. It's what gives each protein its unique job. Okay, so if proteins are the workers, where do they get all their energy? What's paying the bills? Well, that brings us to our next family of molecules, carbohydrates. Think of these as the primary energy currency for pretty much all cellular work. A classic example of this is starch, which is how plants store massive amounts of energy. You see, its building blocks actually link up to form these cool helical structures, almost like a coiled spring, which is just a super efficient way to pack away fuel. And that stored energy is what powers a huge chunk of life on Earth. All right, so we've got our workers, the proteins, and we've got our energy source, the carbs. But who's in charge? Where are the instructions coming from? Well, that brings us to the master blueprint for all of life. You guessed it, the one, the only DNA. To really get DNA, you got to start with its basic building block, which is something called a nucleotide. And each one is actually pretty simple. It's made of just 3 little parts, a sugar molecule, a phosphate group, and this really critical piece called a nitrogenous base. Okay, and here's a little detail that trips a lot of people up, but it's super important. If you just have the sugar and the base stuck together, that's called a nucleoside. But the moment you add that phosphate group, boom, it becomes a nucleotide. That phosphate is the key because it's what allows these units to link up into those famous long chains. So how does DNA store information so perfectly? Well, the secret is all in the bases and this one simple, beautiful rule. It's called Chargaff's rule. It just says that the base adenine, or A, will only ever pair up with thymine, or T, and guanine, G, will only ever pair up with cytosine, C. That's it. This super strict pairing is the entire foundation for how life copies itself. And all of this comes together in that iconic shape you've seen a million times, the Watson-Crick model. First, you get the sugars and phosphates linking up to form 2 long backbones. They're like the rails of a ladder. Then, the base pairs connect across the middle to form the rungs. Now get this, A and T are held together by 2 hydrogen bonds, while G and C are held by a slightly stronger connection of 3 hydrogen bonds. And finally, this whole ladder gets a twist, forming that elegant right-handed double helix. This structure is pure genius. It's the key to both storing information and copying it perfectly. Okay, this is where it all comes together. We have this amazing blueprint in our DNA, and we have the protein workers ready to go. So, how does the cell actually read the blueprint to build the workers? This process is so fundamental to biology that it's called the central dogma. It's actually a pretty elegant 3-step flow. First, the master blueprint, the DNA, stays tucked away safely inside the cell's nucleus. When a certain protein is needed, a temporary working copy called RNA is made. That's transcription. Then that RNA message travels out of the nucleus and is used as a template to build the final protein. That's translation, from blueprint to action.
And this is the part that just blows my mind every time. This entire system, this flow of information from DNA to RNA to protein is the same in you, it's the same in a mushroom, in a fly, and an oak tree. This molecular language is universal across almost all known life. So that leaves us with this one big, incredible thought to chew on. If all life, in all of its breathtaking diversity, is written in the exact same chemical language, using the same grammar and the same alphabet, what does that tell us about our deeply interconnected, shared story?
All right, everyone, welcome. Today, we're going to jump right in and deconstruct one of the most essential pieces of life itself, carbohydrates. It's a word we hear all the time, right? Carbs this, carbs that, but what are they, really? Let's break it all down.
So let me start with a question for you. What could a spoonful of sugar, a potato, and a giant tree trunk possibly have in common? I know, it sounds like a weird riddle, doesn't it? But the answer is actually the key to this whole explainer. All three are built from the exact same thing, carbohydrates. But how? How can one basic material be sweet, another one starchy, and a third one tough enough to build a house with? That's the mystery we're going to solve.
To really get how carbs can be so incredibly diverse, we first need to look at what they are on a chemical level. And believe it or not, the name itself gives us a massive clue. The name carbohydrate literally means hydrates of carbon. And that's because, way back when, chemists saw their formula, which often looks like carbon atoms attached to water molecules. Now, for you chemistry buffs out there, the more precise definition is that they're polyhydroxyaldehydes or ketones. But for the rest of us, what that really means is they're just molecules with two main features, a bunch of alcohol groups and one special, super reactive group that makes each sugar totally unique. You know, every massive structure, whether it's a skyscraper or a complex molecule, starts with simple building blocks. And for carbohydrates, that basic unit, that first Lego brick, is called a monosaccharide. Monosaccharides are just what they sound like, single sugar units. They're the simplest carbs you can get. You literally can't break them down into smaller sugars. They are the absolute foundation for everything else we're about to build. And hey, you know these guys better than you think. Glucose? That's the main fuel that powers pretty much every cell in your body. Fructose is that delicious sweetness you get from fruit. Galactose is a part of the sugar found in milk. And ribose? Well, that one is a critical piece of RNA, one of the most fundamental molecules for life itself. Okay, so we've got our single bricks. What happens when we start to click them together? Well, that's when we start to form much more complex and interesting structures. Let's see how that works. So when you link anywhere from 2 to 10 of these monosaccharides, you get what's called an oligosaccharide. The ones we run into most often are the disaccharides, which, you guessed it, are made of exactly 2 sugar units bonded together. And this is where it gets really cool, because it completely deconstructs the sugars we see every day. The plain old table sugar you put in your coffee, that's sucrose, which is just one glucose molecule linked to one fructose molecule. The lactose in milk is just glucose linked to galactose. And maltose, which you find in grains, is even simpler. It's just 2 glucose units stuck together. But that brings up a really important question. It's not just what bricks you use, but how you stick them together, right? So does the way these sugars connect actually change anything? Oh, you bet it does. A tiny difference in their chemical bond can have a huge impact. So this brings us to this idea of reducing versus non-reducing sugars. It sounds a little technical, but the idea is simple. Most of these simple sugars, all the single units, plus lactose and maltose, have a chemically reactive end that's just open and ready to react. But with sucrose, the reactive parts of both the glucose and the fructose are used to form the bond. They're locked up, so to speak. This makes sucrose a non-reducing sugar, and it's a property that actually makes it unique among these common sugars. Okay, we've gone from 1 sugar to 2. Now let's scale things up. I mean, way up. When you start linking hundreds or even thousands of these monosaccharides together in long chains, you get polysaccharides. These are the true megastructures of the carbohydrate world. And these giant molecules pretty much fall into 2 main camps based on their job. Some of them are like cellular battery packs. Their whole purpose is to store energy for later. And the others? They're like cellular scaffolding, used to build strong, stable structures. So, for energy storage, plants use starch. Its structure is this long, coiling helix. And fun fact, that coil shape is perfect for trapping iodine molecules, which is why you get that classic blue-black color in a starch test.
Animals, like us, use something called glycogen. People sometimes call it animal starch because it's very similar, but it has a lot more branches. That branch structure lets us release glucose super quickly when we need a sudden burst of energy.
And on the structural side, we have cellulose. And get this, it's the single most abundant organic polymer on the entire planet. It's what gives plants their strength and rigidity. Then you've got chitin, which is another incredibly tough polysaccharide that forms the hard outer shells of insects and crabs, and even the cell walls of mushrooms.
So, how does all this chemistry actually matter to us, to our bodies? Well, it all comes down to what we can and can't eat, and this is where it all comes together.
Let's go all the way back to that first riddle. A potato is packed with starch. A piece of paper is made of cellulose. But chemically, they are both just long chains of the exact same building block, glucose. So why is one of them food and the other is, well, not?
The answer is so small it's almost hard to believe. It all comes down to a tiny, tiny flip in the orientation of the glucose building block. See, starch is a polymer of alpha glucose. Cellulose is a polymer of beta glucose. That one subtle difference creates a totally different kind of bond, which leads to a completely different 3D shape. Starch coils up into a floppy helix, while cellulose forms these rigid, straight fibers.
You can think of it like this. That alpha linkage in starch lets it coiled up, making it perfect for packing away energy that our bodies can get to easily. But the beta linkage in cellulose forces the chains to line up in these flat, super strong sheets, which is perfect for building the wall of a plant cell, but impossible for our bodies to break apart.
Here's what's happening inside your body. Our digestive enzymes are like these tiny molecular scissors, and they are shaped perfectly to snip the alpha bonds in starch. That releases all the glucose for energy. But those same scissors just don't have the right shape to cut the beta bonds in cellulose. We just don't have the specific enzyme for that job. So when we eat cellulose, or fiber, it just passes right on through.
From simple sugar to energy to the very structure of the world around us, carbohydrates are just a masterclass in molecular engineering. It's incredible, isn't it? A simple flip in one single bond is the difference between food and wood. It really makes you wonder, what other tiny, subtle changes like that are out there, quietly shaping our entire world in ways...
All right, let's dive deep into the microscopic world. Today, we're going to talk about the molecules that literally build and run our bodies. I'm talking about proteins and their superpowered cousins, enzymes.
So here's the game plan. We're going to start with the absolute basics, what I like to call the alphabet of life. Then, we'll see how that alphabet builds these incredible, complex structures. After that, we'll get into the catalysts that make life happen at lightning speed. And finally, we'll meet the essential little helpers that make it all possible.
So let's kick things off with a huge question. What exactly are these master molecules? I mean, think about it. They're the architects and the construction workers, the messengers and the machines, all rolled into one incredible package. Well, the answer is proteins, and they themselves are built from an even simpler set of building blocks.
And that leads us right into our first section, the alphabet of life. We're about to get up close and personal with the fundamental units called amino acids. Okay, so every single amino acid has the same core structure you see here. There's a central carbon atom with 4 things attached. Now 3 of those are always the same. But that 4th one, the R group, that's the secret sauce. That's what makes each one unique. Just think about it. Our 26-letter alphabet can create endless words, right? In the same way, the 20 different types of amino acids, each with its own special R group, combine to build a practically limitless variety of proteins.
Now, our bodies are pretty amazing, but they can't make all 20 of these amino acids from scratch. The ones we can make are called non-essential. But the others, the essential ones, we're talking about guys like lysine, valine, and tryptophan, we absolutely have to get them from the food we eat. This is why dietary protein isn't just a good idea, it's the only way we get these critical building blocks.
Amino acids also have this really cool chemical trick up their sleeve. They're amphoteric. All that means is they can act like an acid or a base depending on their surroundings. This chemical chameleon ability is absolutely vital for them to work properly inside our cells, where the pH levels can change all the time.
So let's move to our next section and see how this all comes together. We have our alphabet, right? So how do we actually use it to spell out the words? How do we get from these simple amino acids to the complex, functional proteins that do all the work?
Well, it all starts in these tiny protein factories inside our cells called ribosomes. They grab individual amino acids and link them together,
one after another, using this incredibly strong connection called a peptide bond. And what you end up with is a long linear chain, basically a string of beads called a polypeptide. This is a protein in its most basic form.
But here's the thing, a simple chain can't do the really complex jobs. The real magic is in the folding. That initial sequence, that's the primary structure. Then, parts of that chain start to twist into spirals called alpha helices, or fold into pleated beta sheets. That's the secondary structure. Then, the entire chain folds up on itself into a unique 3D shape, the tertiary structure. And that's where the function really begins. For some proteins, it goes one step further, with multiple chains coming together to form a massive final complex, which we call the quaternary structure. And speaking of structure, let's talk about an absolute superstar, collagen. This is the single most abundant protein in the entire animal kingdom. It's the main structural protein in your skin, your bones, your connective tissues. It is literally the molecular scaffolding that holds you together.
But if we zoom out, really zoom out, to the entire planet, the undisputed champion is a protein called rubisco. This is an enzyme found in plants, and its job is to capture carbon dioxide from the atmosphere during photosynthesis. Without rubisco, life on Earth as we know it, well, it just wouldn't exist. It's that important.
The fact that rubisco is an enzyme is actually the perfect segue. Let's really focus now on one of the most vital jobs that proteins have, acting as the biological catalysts that make all of life's chemistry possible.
So what even is an enzyme? You can think of it as a catalyst made by a living thing. The vast majority of them are what we call globular, or kind of ball-shaped proteins. Their one job is to take a chemical reaction that might normally take years and make it happen in a split second. And here's a little tip. You can often spot them by their names, which usually end in ase, like sucrase or protease.
So how do they pull off this incredible speed boost? They don't just add more energy. Instead, they lower the energy barrier, what scientists call the activation energy that a reaction has to get over to start. Imagine you have to get a boulder over a huge mountain. An enzyme doesn't push the boulder harder, it just builds a tunnel straight through the mountain. It makes the journey way easier and faster. But enzymes are pretty sensitive. They're picky, and their activity is massively influenced by their environment. As you can see here, three things are absolutely critical: temperature, pH, and how much stuff they have to work on, which we call the substrate. See, every enzyme has a perfect, narrow little range for temperature and pH where it works at its best. Go outside that range and its performance just plummets. And yeah, more substrate makes it work faster, but only up to a point, where it gets saturated and just can't work any quicker.
Now, enzyme activity can also be turned down, or even switched off completely, by things called inhibitors. A competitive inhibitor is kind of like a key that fits in the lock but won't turn. It just sits there blocking the real key, the substrate, from getting in. A non-competitive inhibitor is a bit sneakier. It binds to a totally different part of the enzyme, and in doing so, it changes the enzyme's shape so the lock itself doesn't work anymore.
For our final piece of the puzzle, we need to look at a really crucial detail. See, many enzymes can't actually do their job all by themselves. They need a non-protein partner to get activated. So let's meet the cofactors.
First up, you've got the apoenzyme. Now this is just the protein part of the enzyme. On its own, it's completely inactive. It's like a car with a full tank of gas, but no key in the ignition. All the parts are there, but it's not going anywhere.
The key, in our analogy, is the cofactor. This is a non-protein molecule. It could be a simple metal ion or something more complex that binds to the apoenzyme and basically turns it on.
And when you put them together, you get the whole functional unit. The apoenzyme plus the cofactor equals the holoenzyme. That's it. That's the active, ready-to-get-to-work enzyme. It's a simple equation, but it's an incredibly powerful partnership.
And these little helpers, these cofactors, they come in a few different flavors. You have prosthetic groups, which are organic molecules that are bound on super tightly, like the heme group you find in the enzyme catalase. Then there are coenzymes. They're also organic, but they only bind loosely, just for a moment while the reaction happens. A lot of vitamins are actually precursors to coenzymes like NAD and NADP. And finally, sometimes all an enzyme needs is a simple metal ion, like the zinc ion that carboxypeptidase needs to do its job.
So we've seen how proteins are built from this 20-letter alphabet, folded into these unbelievably specific machines that run the entire chemistry of life. It is a system of just breathtaking elegance. And that leaves us with one final and a really critical question.
if this entire beautiful system depends on a perfect sequence? Well, all right, let's jump right in. Think about every living thing you can imagine for a second, from the tiniest microbe to the biggest blue whale, and yep, that includes you and me. We're all built from the exact same fundamental set of chemical parts. These are called biomolecules, and they're literally the universal building blocks of all life.
But let's make this personal. Forget all life for a moment. What about you? I mean, what are you really made of? If we could break you down into your most basic chemical elements, what would we actually find? The answer is, well, it's pretty wild.
Okay, so check this out. When you compare the raw elements in the Earth's crust, dirt and rocks, with the elements inside of us, something amazing pops out. Look at those percentages. Life isn't just using what's lying around. No, it's picky. It actively grabs and concentrates very specific elements, especially carbon and hydrogen. This is the big takeaway. Life, at its very core, is a carbon-based operation.
Which of course leads to the next big question. How in the world do we know this? How do scientists actually peek inside a cell and take a chemical inventory? It sounds super complex, right? But the basic method is surprisingly simple. It's kind of like making a very strange chemical soup.
Here's the play-by-play. You take a piece of living tissue, let's say a piece of liver, and you grind it up with some acid. Now the acid's job is to bust open all the cells, letting all the goodies inside spill out. This gives you a thick slurry, which you then strain through something as simple as cheesecloth. Yeah, really, cheesecloth. It's an almost shockingly simple method that perfectly sorts everything inside a cell.
And the result you get from this one experiment is, well, it's beautiful. You get two totally distinct piles. The liquid that drips through the cheesecloth, that's called the filtrate, or the acid-soluble pool. And the chunky stuff that gets left behind, that's the retentate, the acid-insoluble fraction. So, just like that, you've divided all the chemicals in the cell into two major groups based on one simple thing, their size.
So let's start with what dripped through, the filtrate. Everything in this acid-soluble pool is a micromolecule. These are the little guys, with a molecular weight less than 1,000 daltons. And a dalton is just a unit scientists use to weigh things that are incredibly, incredibly tiny. They're small enough to dissolve and pass right through the filter.
So what are these things? Well, think of them as the alphabet of life. Seriously, we're talking about simple sugars, the amino acids that are going to build proteins, the nucleotides for our DNA, plus a bunch of inorganic ions. These are the basic individual building blocks.
And now for the other crew, the stuff left behind in the retentiate. This is the home of the macromolecules. These guys are the biological heavyweights with molecular weights way over 1,000 daltons. They're just too big to get through the filter. And the most important thing to know is that most of them are polymers. Gigantic chains built by linking those micromolecule monomers together over and over and over again.
So, let's meet these heavyweights. First up, polysaccharides. These are your complex carbs. They're basically just massive chains made by linking hundreds or thousands of simple sugars together. This is how plants store energy as starch, or how they build incredibly strong structures like cellulose, the stuff that gives wood its strength. So they're all about energy and structure.
Next up, we've got proteins, the absolute workhorses of the cell. They are polymers of amino acids. Remember that 20-letter alphabet of micromolecules we talked about? While proteins are the endless words, sentences, and entire novels spelled out with that alphabet. They are the molecular machines that do pretty much everything required to keep you alive.
And finally, the most famous macromolecules of all, nucleic acids. We're talking DNA and RNA. These are the master blueprints, the ultimate instruction manuals for life itself. They're polymers built by linking nucleotides together into these unbelievably long, information-packed chains that tell the cell exactly what to do and when to do it.
Okay, but hold on a second. There's a classic biology puzzle here, a real curveball. Lipids, fats, they are also found in that acid-insoluble macromolecule pile. But here's the catch. Their molecular weight is actually small, way under 1,000 daltons, and they're not true polymers. So, what gives? Why do they end up with the big molecules?
And here is the solution to the lipid paradox. It's not about their size, it's all about their behavior. See, lipids hate water. They're insoluble. So when we grind up the cell membranes, which are made of lipids, they break apart and instantly reform into these tiny water-hating bubbles called vesicles. These vesicles are big enough to get trapped in the filter with the true macromolecules. So lipids end up in the insoluble fraction simply by association. not because they themselves are big. Pretty clever, huh? Now, all of these macromolecules are absolutely vital, but we're going to zoom in on proteins for a minute. Why? Because they perfectly illustrate one of the most important principles in all of biology. Structure is everything. With proteins, their shape isn't just important, their shape is their function. It all starts here, at ground zero, the primary structure. This is simply the unique, one-dimensional sequence of amino acids in the chain, read from one end to the other. Think of it like letters on a page. This sequence is the fundamental information, and it's dictated directly by your DNA.
But a straight line of letters doesn't really do anything. So that chain starts to fold. The secondary structure describes these local repeating patterns. The two most famous are the alpha helix, which looks like a winding spiral staircase, and the beta sheet, which looks kind of like a fan folded back and forth.
Now things get really interesting. The chain keeps folding and twisting back on itself into this complex, three-dimensional globe, often described as a hollow woolen ball. And this is where the magic happens. This final 3D shape, the tertiary structure, is what gives the protein its specific job. It creates the perfect nook or cranny to do its work. Change the shape and you break the machine. It's that simple.
But wait, there's more. We're not always done. Some proteins, like the hemoglobin that carries oxygen in your blood, have a quaternary structure. This is when multiple already folded protein chains come together to form one giant functional complex. Think of it like a team. Hemoglobin has 4 separate chains that have to assemble perfectly to get the job done.
So what do these incredible molecular machines actually do? The short answer? Everything. I mean, look at this list, and this is just a tiny snapshot. They're the collagen that literally holds your skin together. They're the trypsin that digests your food, the insulin that controls your blood sugar, the antibodies that fight off infections. Their diversity of jobs is just staggering.
And just to give you a sense of scale, to really blow your mind for a second, the most abundant protein in the entire animal kingdom is collagen. That structural protein we just mentioned. But the most abundant protein in the entire biosphere, so all of life on Earth, is an enzyme in plants called rubisco. It's the enzyme that grabs carbon dioxide right out of the air. It literally builds life from thin air.
Okay, so let's put a pin in this. What are the big takeaways? 1. Life's chemistry is all about carbon. 2. We can neatly sort all those chemicals into small micromolecules and large macromolecules. 3. Those big macromolecules are polymers, built from smaller monomer units. And 4. For proteins, their complex 3D structure is absolutely critical for their function.
Which leaves us with one final thought. If life itself is written in this incredible chemical language, a language of monomers and polymers, of structure and function, then what amazing, unbelievably complex stories are being told inside your own body right at this.
All right, let's dive right in. Today, we're going to peel back the layers of life itself and look at 2 of the most fundamental molecules that make you, well, you. We're talking about the actual blueprint for life and the very materials used to build the whole structure. It's going to be fascinating. I mean, really think about it for a second. What are you made of? It seems like a simple question, right? But the answer is just incredibly cool and complex. If you could zoom way past your skin, your organs, your cells, you'd find this amazing world built from just a few key types of biological molecules. These are the true fundamental building blocks for every single living thing on this planet.
So, in the grand scheme of things, life really boils down to 4 major classes of these biomolecules. You've probably heard of carbs for quick energy and proteins, which are kind of the workhorses doing all the heavy lifting. But for this explainer, we're going to zero in on the other 2 pillars, the super versatile lipids and the critically important nucleic acids.
Okay, first up, lipids. Now you might just think of them as fats and oils, but oh, they are so much more than that. I like to think of them as the expert contractors of the cellular world. They're the ones building all the boundaries and acting as our body's primary energy banks.
So what's the one thing that ties this huge, diverse group of molecules together? It all comes down to their relationship with water. The one single rule that defines a lipid is that it does not, under any circumstances, dissolve in water. They're hydrophobic. And believe it or not, this one simple property is the key to pretty much everything they do in your body.
Now, the basic building bricks for most of these lipids are things called fatty acids, and their shape dictates what they do.
You've got saturated fats like palmitic acid. See how their chains are all straight? That lets them pack together really tightly, which is why they're solid like a stick of butter. On the flip side, you have unsaturated fats like oleic acid. They have these little kinks in their chains because of some double bonds. This kinkiness prevents them from packing together, and that's why they're usually liquid oils at room temperature.
So, how do we actually build one of the most common lipids out there? It's pretty straightforward. You start with the single glycerol molecule. Think of it as a backbone. Then, one by one, you attach 3 fatty acid chains to it. And voila, you get a triglyceride. This molecule is the main, incredibly efficient way our bodies store energy for the long haul.
But just locking away energy is really only the beginning of the story for lipids. I mean, if that's all they did, they'd still be super important. But their most critical role is actually something completely different. It's structural.
And here is where it gets really cool. When you take a lipid and you add a phosphate group to it, you create something brand new, a phospholipid. This tiny change gives the molecule a kind of split personality. It now has a head that loves water and a tail that fears it. And this dual nature is exactly what allows them to form the very boundary of life itself, the cell membrane.
So let's wrap up part 1. Lipids are absolute powerhouses, right? They store energy, they provide insulation, they form the membranes of every single one of your cells, and they're even the starting point for critical hormones. For instance, cholesterol gets a bad rap, but it's the precursor for vitamin D and hormones like testosterone. While our bodies can make most of the fats we need, there are a few essential fatty acids like linoleic and arachidonic acid that we absolutely have to get from our diet.
Okay, time to shift gears. If lipids are the builders and the banks, then nucleic acids are the architects and the librarians. These molecules hold the master plan, the complete instructions for everything a cell is and everything it needs to do. So what are nucleic acids, really? Well, molecules like DNA and RNA are what we call polymers. That's just a fancy word for a really long chain made up of repeating units, kind of like beads on a string. Their entire job, their whole purpose for existing, is to store, read, and pass on the genetic blueprint of an organism from one generation to the next.
And what are those repeating units I just mentioned? They're called nucleotides. Each one is a simple little 3-part structure. First, you've got a 5-carbon sugar, either deoxyribose or ribose. Then there's a phosphate group, which acts as the connector. And finally, you have a nitrogenous base. This last part, this is the part that carries the actual code. These nitrogenous bases are literally the alphabet of the genetic code, and they come in 2 flavors. You've got the purines, adenine and guanine, which are a bit bigger with 2 rings, and then you have the smaller, single-ringed pyrimidines. Now, the key detail to spot here is the difference between thymine, which you only find in DNA, and uracil, which is its replacement in RNA.
When you put the 2 stars of the show, DNA and RNA, side by side, you see some really clear differences. DNA is the master blueprint. With its deoxyribose sugar and that iconic, stable double helix structure, it's literally built to last for long-term storage. RNA, on the other hand, is more like a working copy or a messenger. It uses a different sugar, ribose, it swaps out thymine for uracil, and it's usually just a single strand. This makes it way less stable, which is actually perfect for its job of carrying short-term messages.
So, let's bring this all home. We've looked at the boundary-building lipids, and we've looked at the information-carrying nucleic acids. But how do these 2 completely different things come together to create, well, you?
It really is as simple and as profound as this. The nucleic acids, your DNA, contain the complete set of instructions for building everything. And the lipids, especially those phospholipids, form the physical structures, like the walls of your cells, that actually house all of the machinery of life and allow those instructions to be carried out. One is the blueprint, the other is the building itself.
And that leaves us with one last, kind of mind-blowing thought. We're now at a point where we understand this code and these building materials so incredibly well that we're actually learning how to edit them directly. So if DNA really is the blueprint for life, what happens when we start to rewrite that code? We're not just changing a molecule anymore, we're changing the very definition of what's possible for life itself. Thanks for joining me.